Here we go.
There it is.
Hey, everyone.
Hello.
Sorry for the delay.
No worries. Well, it's 11:01. Do you want to go ahead and start or? Yep.
That sounds great. Yeah.
Okay. So welcome everyone for the webinar series on aging clocks. My name is Young Oh, and I'm a program officer at NHLBI in the Division of Cardiovascular Sciences. And today's event is part of the series of webinars developed by NIH Geoscience Interest Group, or GSIG. The GSIG actually started about 10 years ago, 2012, in order to advance and highlight the burgeoning field of geoscience, which is a translational field that exists at the intersection of the basic biology of aging and the clinical development of interventions that could improve human health at all the ages. GSIG members come from more than 20 NIH institute and centers and have a shared interest in the impact of aging on various human diseases and conditions. Our group is very interested in learning about measures of a biological aging and the latest advances with aging clocks. Thus, we started this several the webinar series, including those based upon epigenetic as well as proteomics data. During today's webinar, if you have any questions for the speaker, please submit through those through the Q&A box, and we'll read them as many as possible during the discussion period at the end of the talk. As you know, we have launched the first webinar series successfully last month, and today we have two speakers, and we're going to talk about different aging clocks, specifically about comparing first and second generation epigenetic clocks and their potential applications. Each speaker have about 30 minutes, and then we'll try to reserve about 20 to 30 minutes at the end of the talk. Or I was told, if you have a burning question after each speaker, you can first speaker, for example, then you can we can entertain a few questions, but majority of discussion will be held at the end of the talk. So we have two speakers, as I said. First, Dr. Daniel Belsky. Dr. Daniel Belsky is an associate professor of epidemiology, Columbia University, Malman School of Public Health, and he's also a faculty member in the Robert N. Bertler Columbia Aging Center. Dr. Belsky's research combines public health, population sciences, and genomics. He is interested in understanding how genes and environments combine to share health across the life course. With Terry Moffitt and Ashlam Caspi, Dr. Belsky developed the pace of aging method to quantify the aging process from longitudinal analysis of human physiology. Dr. Belsky is the principal investigator on several NIH-funded projects, including research on the Dutch hunger winter family studies, the Kelly trial, and also My Goals for Healthy Aging multi-center randomized controlled trial. And our second speaker will be Rav Segal. And Rav is actually a PhD student at Yale University in Dr. Alva Higgins Chan Laboratory, and he's studying computational biology and human aging. Raghav is a Glover Grant Awardee and Impetus Aging Grant Awardee, and a Yale Center Biology Program Fellow. Obviously, he's a rising star in this field. Raghav research harnesses multi-omic complex data to ask questions about aging, including what biological systems drive aging, and how does aging in these different biological systems and processes give rise to diseases such as cancer, diabetes, and Alzheimer's. So, without further ado, I'm gonna let our first speaker, Dr. Belsky, talk about his research and interest. Go ahead.
All right, so here we go. I'm going to share my slides. This is me. This is my disclosure. And this is my first slide. So let me just say, as I dig in, Please feel free to interrupt me as I go along and ask questions. We're a very small group, and so it may be. I think we can be pretty casual about this. So this is where I begin in all of this, what I've learned from Ron and others. This is my cartoon version of the geroscience hypothesis that aging begins with an accumulation of cellular level changes that feed forward to drive decline in system integrity, causing functional decline, disease, disability, and mortality. And of course, what makes it the geroscience hypothesis is that we believe that intervention to slow or reverse the accumulation of these molecular changes can delay or possibly even prevent these declines in the functioning and resilience capacity of our organ systems and tissues, and ultimately forestall functional decline, delay disease onset, give us more years of healthy life. This is another cartoon illustrating, roughly what we see from a clinical perspective about human health and aging, with the x-axis representing aging from midlife into later life, and the y-axis representing some measure of health, with the blue and red sections of the diagram distinguishing what the clinician will see as a healthy patient, a disease-free patient, and the red part, the diseased patient. But obviously, health is some kind of a continuum. And the idea is that we, in midlife or young adulthood, see a population with relatively undifferentiated health at the phenotypic level, but in whom reside molecular differences that will ultimately guide diverging trajectories of aging-related changes in health, illustrated by the light blue line. That's our kind of prolonged health span case, the darker blue line, and then the red line where people are experiencing accelerated decline. And the purpose of illustrating the aging process in this way is to highlight the early stage of this decline process in midlife as the optimal setting for preventive intervention. So if we want to deploy geroprotective therapies to delay or prevent disease, we need to do so before people are getting sick. And that means that we need to be intervening relatively early in the life course. There are obviously applications of geroprotectors that are curative, and that we can imagine delivering later, but for actually changing human health span, intervention has to start early. And I think you could make the case it needs to start much earlier than this. But this is when I think we can imagine trials of preventive geroprotective therapies. Of course, if we imagine such a trial, we have the challenge that the outcomes of that trial are not observable for many decades. And neither our biomedical research apparatus or our researchers themselves or their patients can really afford to wait that long. And so what we're ultimately after are metrics that can, in the course of a trial running for months or years, read out on the impact of the geroprotector on this underlying pace of biological aging. So that's what we need. And in this, again, this stylized diagram, what we're suggesting is the pace of aging is going to remain constant from baseline to follow-up. And in the treated group, we're going to see some reduction in the pace of aging or slowing of the aging rate. The idea of quantifying biological aging as an underlying cause of disease is not new, and I think it's been around in the field of gerontology and geriatrics for some time in the form of indices of frailty or deficit accumulation, and we could think of multimorbidity as being a flavor of one of those. But these kinds of indices are not particularly variable in the phase of the life course. I'm arguing we need to turn our attention to. They don't identify risk until after deficits are already established. And as a consequence, they are relatively insensitive to change from intervention. So it's obviously significant if we can cure diseases or repair disabilities. But that's a much heavier lift, so to speak, for the therapies that we're trialing. And if what we're trying to do is slow aging to prevent disease, these may not be adequately sensitive. And so where the field has moved in the past couple of decades is from this identification of deficits and quantitation of their accumulation toward approaches that are targeted at these earlier phases in that geroscience cartoon that I introduced earlier, either at the tissue and organ level or at the molecular level. And so you can see in the pictures different types of assessments that might be conducted. But in these data-driven approaches, rather than presuming we know what to measure, we define phenotypic surrogates of the aging process and then mine data on organs and tissues or cells and intracellular processes or products, and then use machine learning to devise algorithms that summarize that phenotype from the data on the organ and tissue or molecular substrates. And methods, there are a range of methods to do this. I learned how to do this in the beginning, in fact, from Regev's earlier mentor, Morgan Levine, and we've all learned a lot from Morgan's subsequent mentor, Steve Horvath, and many others. But metrics devised via these methods have a number of properties that I think recommend them to the attention of the Geroscience Interest Group and have certainly directed my research program. And those are that they're variable in young people. They're variable in midlife, they're variable in young adults. In some cases, they're even variable in children, although I think we don't yet know enough to make strong interpretations of them at that early age. They provide preclinical assessments of risk. So measurements of these omics-based or multi-analyte-based indices of biological aging are predictive of the future onset of disease and disability among individuals who appear clinically to be healthy. So they differentiate a population who looks homogeneous to clinical medicine. And then finally, we hypothesize and have some evidence that they may be more sensitive to these early interventions than conventional metrics of deficit accumulation. And there's some nice reviews of developments in this field. Last year, the Weiss-Corey lab has a paper in Nature Review Genetics. And then this year, Madi Mokri, working with the Gladyshev lab, have a lovely paper out in Cell summarizing a lot of what's gone on. And for those of you who downloaded the slides, you can click the links and find the papers. So I'm going to spend the rest of my time talking about a particular flavor of these data-driven metrics of biological aging called epigenetic clocks. You're all familiar. That's the title of today's session. And I'm not going to sort of dive too deep into the biology of DNA methylation, not least because I'm not a card-carrying geneticist, but also because the way we study DNA methylation in the context of epigenetic clock research, at least today, is in some ways abiological. We're not thinking about the mechanistic function of these chemical tags that bond to our DNA sequence and may recruit chromatin to enclose the DNA and inhibit transcription. We're instead analyzing them as sort of black box repositories of information about our biology. And that comes with some strengths, but also some pretty important limitations that we'll come around to as I get to the end of my, my slides. And there's a, the figure there is from Steve's 2013 paper in Genome Biology that kind of blew this field wide open. And I'll also just direct you to his 2018 Nature Reviews Genetics article where he kind of walks through the genesis of the epigenetic clock field and some of what his thinking about what it means look like. So in the beginning, clocks were developed using what I think is a very intuitive approach, but one in fact that has proved not terribly helpful to the research that I do, or I think the research that many people in the public health and clinical space do. And that is, they simply fit variation in the blood methylome to variation in chronological age. So they compiled samples of individuals who varied in the age at which their blood was sampled. They extracted DNA from the whole blood. And then they used a relatively simple machine learning technology called elastic net regression to devise predictive algorithms of chronological age. And then they did the thing that makes this all kind of interesting, which is they asserted that the errors in those predictions were information about how fast a person was aging. And that's illustrated in this slide from Bernice Benayoun's paper. And so what you see here is a age on the x-axis and biological age on the y-axis, and there's a diagonal suggesting a perfect correspondence. So in some idealized reference population, your chronological age and your biological age are on average equal. But obviously people vary. and that's what the red X's illustrate, with some people above that diagonal exhibiting an accelerated pattern of aging. So they're older than we expect them to be based on their birth certificate, their chronological age. And those below the line are younger than we expect them to be. And the silhouette figures kind of argue that we should think of these predictions that are older than expectation as signaling progress toward frailty, disease, and death, and the ones that are younger signaling resilience or resistance to the aging process. But empirically, we actually find that these first generation epigenetic clocks are at best weak and inconsistent predictors of morbidity and mortality in free living humans. There's somewhat better evidence in animal studies, but that's not my expertise, and so I'm not going to dig into that. But I think we can imagine why building a model like this in a laboratory animal that has a less genetic variation, sometimes no genetic variation, and consistent environmental conditions might work less well than when we do it in humans. So we and others published data suggesting that these first generation clocks were not as good at this job of measuring biological aging as they were cracked up to be. And to their credit, Morgan, and now working with Steve, went back to the drawing board and came up with a new method. I got ahead of myself there. Let me step back for a second. Let me tell you why these clocks are, maybe not as good as we'd like them to be. And that will help set up the innovation that Morgan introduced. So the guy in the figure there is Abraham Wald, who was a statistician at Columbia, and he led a team that was tasked with figuring out where we should add armor to bombers that were running missions out of England into Germany during the Second World War. And the figure next to Abraham illustrates what his team came back with. And what you're seeing in those red dots is those are the spots on the plane where planes returning from bombing missions came back with bullet holes in them. And Abraham and his team then recommended to the Army that they put armor where the bullet holes weren't, because they had the realization that the reason there weren't any bullet holes in those other parts of the plane was that the planes that had bullet holes in those parts didn't come back. And so that has implications for the way we think about studying human aging. So if you look down on the slide, you can see a figure illustrating survival curves for different populations within the United States. And what you can see there is that by the time you get into people's 70s and 80s, there's a lot less of the population left than there is if you were to sample people in their 20s and 30s. And so this approach of sampling individuals across the age spectrum and comparing the differences between them and asserting that differences in the biology between young people and old people are the biology of aging brings with it the confound that whereas the young people are drawn from the full population, the older people are drawn only from the survivors. And what differentiates their, in this case, blood epigenome, may be signs of aging-related damage, but they may also be signs of resilience to that damage. These are the successful agers in our population, and so they may not be a good model of what the parts of aging we'd like to prevent should look like. So there it is on the slide. The second piece of this is something that Terry Moffat has talked a lot about, and I know has addressed this group about, which are cohort effects. And so in a cross-section of data, people who differ in their age also differ in the point in history when they were born. And when in history you were born has significant consequences for the exposure regime under which you grow, develop, and live. And that exposure regime will have consequences for your health, your survival, but also your blood methylome. So this is a figure from a paper in Nature Reviews Neurology, but it illustrates some of the differences in the life histories of people born at different times. And the consequences, of course, that the signal that we refine from the methylome could be aging, or it could be any of these other exposures that are patterned across historical time. So now to Morgan's innovation, and then further refinement from Steve and his colleague Ake Liu, they flipped the model around. So instead of asking if your DNA knows how long you've been alive, they instead asked whether your DNA knows how long you're going to stay alive. So they compiled methylomes from individuals in studies that had follow-up to ascertain survival time, and then they modeled survival time as a function of the methylome, again, with the premise that people whose predicted hazard of mortality was greater than expectation, were evidencing a faster process of aging, and those whose predicted hazard of mortality was below expectation were aging more slowly. And so how did they get the expectation of what your mortality hazard should be? In the case of Morgan's work, where this is more clearly articulated, what they did was they modeled the mortality hazard as a function purely of chronological age and used that as their reference. And that's also how you convert the mortality hazard into a biological age estimate. And Stephen Ake did something similar. And in the case of both PhenoAge and GrimAge, these second generation epigenetic clocks built to predict survival time, there were physiological intermediates. So in Morgan's case, she first built an algorithm to predict death from blood chemistry data and then trained the methylome to predict that. In the case of the GrimAge, they first built algorithms to predict blood protein levels from the methylome and then trained those DNA methylation predictions of blood proteins to predict mortality. But I think it's simpler if we just think of these as clocks trained to predict survival time. So the survival time clocks were much better at predicting morbidity and mortality, and GrimAge in particular, and its second generation instantiation, GrimAge 2, are quite strong from the perspective of single biomarker predictions of mortality. But they come with the limitation that they are silent about when the differences that they identify between people were accumulated. So the nature of these models is such that they're summarizing the aging that has occurred across a person's entire lifespan. And so we don't know whether the differences between individuals and their GrimAge reflect differences established at the very beginnings of life or at some particular moment in their life course, or in fact reflect the gradual accumulation of change that we think is the basis of biological aging. And so here on the right-hand side of the slide, you see a figure from a 2019 paper in Genome Biology illustrating how age-associated, chronological age-associated changes in the methylome are quite rapid in the first couple of decades of life and then slower later on. So that may lead us to suspect that some of the variation we see between people in these clocks is really about early life stuff and not so much about progressive accumulation of damage over time. At the bottom of the slide, I'm illustrating some of the implications of this measurement strategy for the sensitivity of these instruments in the context of clinical trials. And the point I'm trying to make here is that if an epigenetic clock is trained to summarize lifelong aging, then most of the information it contains is not about the period of the life course covered by a clinical trial. So there's a lot of aging that happens, only a little bit of it happens during the trial. If in fact your intervention slows aging but doesn't reverse it, the clock itself may not be able to pick that up. So it was in response to these limitations of the existing clocks that we set about trying to develop a complement, which is Dunedin pace. And so the Dunedin pace clock is different from the second generation clocks in that rather than measuring how much aging a person has experienced, it's designed to measure how fast aging is happening right now. It's we use the metaphor of a car. We can think of the first and second generation clocks as odometer measures of aging. They tell you about distance traveled. Dunedin pace is designed to be a speedometer measure of aging. It tells you how fast change is occurring. And so what you see in the graph is this illustration of the idea that you might in fact see variable pace of aging resulting in differences in system integrity over time. And this is work that evolved over about 7 or 8 years in collaboration with Terry Moffat and Avshalom Caspi, and more recently, David Corcoran and Karen Sugden. We began by modeling changes in people's bodies over time. Because we were motivated by the idea that if aging is this progressive decline in system integrity, then we can infer the rate of aging from decline occurring across multiple systems. And if we focus on a population not yet affected by disease, this will refine our observation of aging as a process distinct from any individual disease process. And so this was our 2015 paper in which we accumulated data on a cohort that Terry and Avshalom had been following for decades at that point. They were 38 years old. The cohort has been followed since birth. But we had 3 time points of measurement spanning 12 years at which we were able to harmonize 18 different measurements of the functioning of different organ systems in the body. And so we modeled changes in each of those organ systems individually. For each person, we quantified how much faster or slower that organ system was changing than the cohort norm. And then we compiled those rate of change estimates into a single index that was scaled to have a mean of 1. Separately in men and women, we allowed men and women to exhibit different patterns of aging. And scaled by that mean such that the pace of aging itself is denominated in units of years of physiological decline per years of calendar follow-up. And so what you can see in I guess the data on this slide in the middle may be a little bit confusing, but there are the graphs drawn for each biomarker, and they're all scaled so that higher values correspond to the what we expect the older value to look like. So for things that decline with aging, like lung function or telomere length, they've been reverse scored so that the upward, the positive manifold in the graph is illustrating that all these systems are mostly moving in the direction we expect, even in these young, healthy people, with the exception of that lipid ratio. So when it came to building this next generation epigenetic clock to measure the pace of aging, we used Dunedin pace as the dependent variable. So we replaced what in the first generation clocks was time since birth, and in the second generation clocks was time until death, with, in this pace of aging clock, the rate of decline in organ system integrity. Forgive me, my building is trying to conserve power, so it thinks when I don't move enough, I'm not here anymore. So we took a stab at this back in 2020, but we didn't really know what we were doing, including, in fact, when it came to the name of the index, which we called Dunedin POAM, not a piece of IKEA furniture. Although we didn't really know what we were doing, the clock that we built actually showed some signs of progress. It was associated with morbidity and mortality in samples of older adults. It was associated with histories of adversity in younger adults. And it was associated in similar magnitude and direction with indices of functional performance as the original pace of aging itself. So a couple of years later, and some more knowledge and water under the bridge, we came back to the problem with better methods, better data, and a better name. And so Dunedin pace, which is the algorithm out there and available to all of your researchers, is a measure built in the same fashion, but based on 4 time points of follow-up data, and using a refined approach for curating the DNA methylation data that we're feeding into the elastic net regression. Again, just to sort of hammer on the point, because the Dunedin study is following people up from young adulthood through midlife, we're not affected by survival bias. Nobody's died of aging-related disease by the age of 45. Because they were all born in the same year, we're not vulnerable to these historical time or cohort effects. And because we're deriving our index from repeated measures, phenotypes of longitudinal change, we know when the variation that we're observing manifests in the life course. And then finally, because we're studying people in midlife, this is sort of the time when we think intervention is likely to be most impactful. One important feature of Dunedin pace is that it was built to be technically reliable, meaning that if you measure it in the same DNA sample twice, you should get basically the same value. And this was built on some observations that Karen Sugden made that recognized that many of the probes on the Illumina arrays that we use to derive these measurements don't have very high technical reliability. And one theory of why that is that those probes simply ought not to be variable in blood, and so whatever measurement variation exists is in fact error. But what you see on the graph here are intraclass correlation coefficients for Dunedin pace in gold, which was derived by machine learning analysis of only the most reliable probes on the Illumina array, as identified by Karen, as compared with our first generation clock, Dunedin POAM, and then the first generation clocks of Horvath and Hannum, and the second generation clocks PhenoAge and GrimAge. And what you can see is that although GrimAge actually looks pretty good, the others don't. And I should just, you may already be familiar, and Regev may cover this, but there's another, in some ways, more elegant solution to this problem of reliability that Morgan's lab introduced a couple of years ago. That Albert's paper in Nature Aging walks through that. So just a couple of observations about Dunedin pace, and I'll get out of your way. One of the things that struck us about Dunedin pace, and this was also true of the first iteration, POAM as well, is that it even though it was developed in a group of people who did not vary in their chronological age, the measurement itself is correlated with chronological age. And that's consistent with demographic expectations about the pace of aging. In fact, biodemographers will quantify the pace of aging of the population as a function of age-dependent change in mortality risk. So we expect that the rate of aging, in fact, accelerates as we grow older in proportion to the accelerating force of mortality, or the Gompertz law. And so what we see here in pace of aging is consistent with that expectation. People who are chronologically older appear to be aging faster. People who are younger are aging more slowly. We also see robust prediction of a range of different aging-related health outcomes that we care about. So I think there's somebody on the call from Heart, Lung, and Blood. You can see that people with faster Dunedin pace are at increased risk for incident cardiovascular disease and incident stroke. And these are data from the Framingham Heart Study offspring cohort. So it's about a dozen years of follow-up from the timing of DNA methylation measurement to the final follow-up for these outcomes. For those of you who may be interested in neurological disorders, we also see robust associations with the onset of dementia in the Framingham Heart Study. And actually, Karen has a paper reporting those data that also covers analysis of the Alzheimer's Disease Neuroimaging Initiative data set, replicating these findings and illustrating that, in fact, Dunedin pace does show these robust associations with risk of dementia. We also know from analysis of other cohorts is associated with cognitive functioning in younger people. It's associated with risk of mortality. You'll notice that in these plots, the grim age, particularly for prediction of heart disease and mortality, is doing quite well. And grim age is, in fact, quite good at predicting incident cardiovascular disease and mortality. But these particular estimates are not super trustworthy because they come out of the Framingham Heart Study, and that's the same cohort grim age was developed in. Just a few more data points. Here you see incident and prevalent chronic disease in the normative aging study on the left, and then incident disability as measured by 3 different ADL indices in the Framingham Heart Study on the right. And there, in fact, it looks like, yeah, so there are many replications of these kinds of observations and more coming out every day. Terry Moffat has compiled a bibliography of many of the studies that report Dunedin pace, but you can also just type Dunedin pace into PubMed or Google Scholar, and you'll find a laundry list of them. So these are sort of illustrative observations. They're not intended to be comprehensive. Another observation that's really critical to my own research program is the observation that Dunedin pace is faster in people that have had harder lives. So people who we know to be at risk for earlier onset of aging-related disease and shorter lifespan as a function of what we call social determinants of health. So here you see data from a cohort of 18-year-olds, and we see that those who were born into families that were poor are aging substantially faster than those who were born into families who were rich on the left. And then you see the same result in the context of history of victimization on the right-hand side. So people are familiar with adverse childhood experiences. What we're illustrating here is essentially that ACEs predict pace. And that's another observation that's been replicated in a number of other data sets. This is a list from TEMI. 51 different cohort studies have now implemented PACE, and a growing number of trials as well. So we will see a lot more data soon. I know I'm sort of a little bit over time here, so I'll just quickly move through the final observation about PACE. We actually have tested PACE in the context of a geroprotector trial. Many of you may be familiar with work on animals in the context of caloric restriction. So the reduction in macronutrient intake with maintenance of micronutrient sufficiency, it's not starvation, and it does make worms, flies, and mice live longer, and it definitely keeps monkeys healthy, although from what I understand, they are mad as hell. The calorie trial was designed to test safety and efficacy of what, from the perspective of mouse researchers, is low-dose caloric restriction over a period of 2 years of time in healthy, non-obese humans at 3 centers in the United States. And what Calorie found was that it's awfully hard to get people to do CR, and in particular to do it at the same level. They're not like mice who you can control feeding for. The middle plot is illustrating variability in CR in the control group, the ad libitum group on the left, and then substantially below target CR in the treatment group on the right. Nevertheless, Calorie-induced substantial and sustained weight loss. And in fact, a number of observations have been made that suggest Calorie did do what it was supposed to, even if it didn't modify its pre-specified endpoint of resting metabolic rate. It did nevertheless cause loss of mass with preservation of muscle, improved cardiometabolic and general physical health, better mood, better sleep, and some provocative signs of slowed biological aging, maybe most provocatively and most recently, or not quite most recently on this slide, the observation of improved thymic function based on analysis of adipose immune cells in a science paper from Deep Dixit's lab. That was the subject of a presidential symposium at GSA last week. So we had initially gone into the Calorie study to analyze their safety lab data to see if we could compose metrics of biological aging that were introduced by Morgan Levine, and found that in fact, Calorie did appear to slow aging as measured by blood chemistry indices of biological age. Using a method proposed by Clemmerant and Dubal, a version based on Morgan's own pheno-age, and then on the far right is a metric introduced by my colleague Alan Cohen. We published this first in 2017. We put out an R package in 2021. That's Dae-Eun Kwon, the author of the R package, on the slide. But we finally got money from NIH to do this analysis with epigenetic clocks, and this is what we saw. So there are also data in the paper on first generation clocks. They're not altered by the intervention. But what I want to draw your attention to here is that the second generation clocks are also not altered by intervention. They tick. People are older at 12 months than they are at baseline. They're older at 24 months than they are at 12 months. But those differences are not systematic with intervention. We would expect to see the red line indicating the CR individuals sitting below the blue line, and we don't. In contrast, what we see for Dunedin pace is that from baseline to 12 months, Dunedin pace is slowed. And then that slowed pace of aging is maintained between 12 and 24 months, consistent with the hypothesis that CR intervention should slow biological aging. It's worth noting there's extraordinary variability in individual responses to this intervention. So while the trends are apparent, you basically have every possible pattern of response. Some have suggested this is due to the genetic heterogeneity or perhaps life history heterogeneity in the humans in the trial. It could also be due to noise in our measurements. And I think there are many interpretations that can be made of the patterns. We can talk about those. So I'll just wrap up here with the observations that these biological clocks are new opportunities for research on aging and perhaps into aging processes as causes or consequences of the diseases that the institutes that some of you come from focus on. I hope you get the sense that the endpoints used to develop these measures have real consequences for their strengths and limitations, and that differences in how old people are maybe a bad place to start if we're trying to devise algorithms that predict differences in health span and lifespan. We think, although we have only really one trial to move us toward this conclusion, that pace of aging measures may be more sensitive to short-term interventions than odometer measures. So use speedometers if you're trying to identify intervention effects. Although if you think your intervention actually reverses the aging process, it may be the case that a more rigorous test would be a second generation, mortality prediction clock. Ultimately, of course, we need to know if intervention effects play forward into differences in health span and lifespan. In the case of the Calorie trial, we're going to get a look at that with follow-up data SciDOS is generating and a 10-year follow-up of the Calorie participants post-intervention. There's a lot to do, and I think Raghav is doing some of it, so you'll hear more about some of the solutions that are coming. But a major challenge of these clocks, of course, is that we don't know what biology they represent. And there are different strategies people are proposing to attack that question. One of them is to data mine more focused phenotypes to try and isolate clocks or clock components that reflect particular biological processes. And another is to curate more biologically informative or interpretable units to feed into the machine learning analysis so that what comes out may be, may have more interpretable biological meaning. There's a lot of robustness testing we need to do, and I think that we have a really good track record with the clocks that are out there now. GrimAge and Dunedin Pace in particular, across different ancestry populations in the US and Europe. But we really don't know much about how these clocks work in what we might call non-weird, weird being Western, educated, industrialized. I forget what the R stands for, and I think D is developed. But the point is that in low and middle income countries, or countries that have very different immunological context than the Western countries where the clocks were developed and have been validated, we I think, shouldn't be confident that we can interpret their meaning. And the same goes for measurements of the clocks taken very early in the life course, where we see variation. Some of the exposure associations are consistent with what we would expect, but we don't yet have studies relating clock variation in children to the kinds of outcomes that would give us confidence that we're actually measuring this aging process. We also are desperately in need of longitudinal repeated measure studies that can say change in the clock means change in health in a meaningful way. And of course, we need more trials to test whether these clocks are actually modifiable. And perhaps with integration across levels of analysis to establish that variation induced in a clock corresponds to variation in other features of aging biology. So I see there's a question in the chat about whether Dunedin Pace is modified by rapamycin, and we don't know yet, but there is a trial ongoing now that we're going to generate some data for that Yushin Su is leading. So we'll have an answer for you in the coming years, and perhaps others will have an answer even faster than that. I want to highlight for you, as I hand off to Raghav, one study just out in the journals of gerontology that is addressing that need for repeated measures longitudinal change analysis of epigenetic clocks in relation to future outcomes. And these are data from the SATSA study. So these are twins in Sweden, they're older, but they have repeated measures data on their methylomes and on a frailty index. And what they show in this study is that changes in Dunedin Pace, although not the other epigenetic clocks, forecast changes in frailty. And that's in epidemiological studies, really a hallmark of the robustness of an association when you can observe it not just in cross-section or not just exposure at time 1, outcome at time 2, but when change in exposure predicts change in outcome, we block a lot more of the confounding that we're concerned about. So this is by no means game over, but it is a positive sign. So this code is publicly available, and that's us, that's more of us. And it looks like I don't have my thank you slide. And so I am sorry, but thank you very much for the opportunity to address this group. Thank you to Ron, thank you to Young for inviting me, and to Siobhan for all of the logistics that made this possible. So maybe we have a couple of minutes for any other questions, or maybe I see it's actually late. We probably need to go right to Rega, but I'll let you take it.
Okay, sounds good. I know there are a few questions came up, but why don't you go right into Raghav and then have the discussion together at the end. Go ahead, Raghav.
And everybody see my screen? I think so, hopefully.
Yes.
Okay, thank you for having me here today. I'm Raghav. I am a PhD student at Yale. Before I go into my presentation, just some conflicts of interest, some of the research that I will present on systems clocks that's been patented and been licensed out to an aging biomarker. I'm a scientific advisor at LTF, and I'm also a scientific advisor at Cadrean Biopharma. With that, I'd like to jump in. So Siobhan was very generous in sort of providing me with some sort of key focus points. And what I've done is I've taken those key focus points and sort of divided them into three broad sections. The first section, which I'm calling a brief history of clocks time, we'll talk a little bit about the similarities and differences between epigenetic aging clocks and the potential advantages and disadvantages of utilizing each of these clocks. Some of this will sort of cover what Dan has already spoken about, so it'll be kind of a repeat, but hopefully that reiteration will be helpful for everyone. And in my second section, which I will talk about which tissue type is ideal to classify and predict age, I kind of give away the answer, which is blood holds the aging secrets within its crimson's depths. And I will tell you why I believe so in that section. And then in the last section, looking into the clock crystal ball, I'll be talking about sort of what are the future research and clinical applications of these epigenetic aging clocks. So without much further ado, I'd like to start into my first section, a brief history of clocks time. And I kind of want to give homage to the fact that almost 50 years ago, we knew that there were some relationships between DNA methylation and age. And so this paper, when I did a quick Google search, I kind of found this paper, which was one of the first papers that showed some relationship between DNA methylation and age. And actually, as way back as 1999, we were already sort of looking at that there is a very high correlation between methylation and chronological age. So this is, in this case, that is the percent estrogen receptor methylation and how that changes with chronological age. And actually, in 2011 is when we kind of tried to build the first predictor of chronological age using DNA methylation data. And that kind of led to what we call as kind of the boom of epigenetic clocks. We got what we call as the first generation clocks, as Dan mentioned, such as the Hannum clock, the Horvath multi-tissue clock, and the telomere length clock. All of them were trying to predict chronological age or some other proxy of chronological age. And then we got sort of the second generation of clocks that started to predict more complicated traits of aging or mortality. Traits included like smoking, obesity, while other clocks like PhenoAge and GrimAge were trying to predict mortality directly using DNA methylation data. And as sort of Dan mentioned about sort of the Gen X clocks, of which Dunedin pace is a part, which started predicting even more sort of better predictors of aging in the sort of form of pace of aging, while some other clocks went deeper and started predicting sort of tissue age, such as PC brain age, and others like single cell clocks. Now there has been a pan-mammalian clock. So there's been a lot of variety of clocks that are out there right now. And the question then comes is, how similar are these clocks? Are these clocks really doing or capturing something different, or are they all capturing the same thing? And if you look at a very basic level, and this is what I'm showing data here is from correlations between these clock scores in the Baltimore longitudinal study of aging. Many of you would be familiar with this study. These clock scores are very highly correlated, and these are just the absolute clock scores. And the obvious answer to that is because all of them are trying to track with age, which is why they are very highly correlated. And that's kind of where the idea of what I call as age deviation, formerly called as age acceleration, comes in, where age deviation is essentially the residual of biological age after adjusting for chronological age. So essentially you draw a simple linear model between biological age predictor and chronological age, and sort of the difference between a particular point from that linear model line is what we call the residual. And this sort of deviation from this model, or age deviation as we call it, has been shown to be associated with a wide variety of aging phenotypes. Rather, this age deviation has been associated with both causes and outcomes of aging. Causes such as sort of low SES, low education, as Dan mentioned, HIV, BMI, so on and so forth, but also outcomes of aging, of course, mortality, but also like cancer, cardiovascular disease, diabetes, so on and so forth. And so when we actually look at the age deviations between these clocks, that's when we really start to see these differences between these clocks. So on the left-hand side is kind of what the correlations are of the absolute clock scores. And on the right-hand side, now you see are the age deviations. Of course, none of them are correlated with age because we have regressed out age from all of these clock scores. And what we see is that generation 1 clocks, the kind of the Horvath, the Hannum, Skin and Blood, Zhang clock, all of them are sort of very highly correlated in some ways. And when you compare that to sort of the second generation clocks, PhenoAge and GrimAge, they seem to be relatively less correlated. I'm not including Dunedin pace here because pace of aging is a completely different metric altogether, and I'll talk about that in a minute as I move forward. So then the question is, which clock is best? And that's where kind of there can be different perspectives on how to think about clocks. I think Dan gives a very impressive insight into how these clocks are trained and what particular biases can come up while they are being trained. The way I look at these about how to sort of differentiate between clocks is really the outcomes they are trying to predict and how good are the outcomes they are trying to predict. And so this is from my own research where we looked at clocks in the Women's Health Initiative, which is a cohort study of about 5K plus individuals. We did a phase stratified analysis, and we looked and associated all of these different generation 1 and generation 2 plus clocks with a wide variety of aging outcomes, such as mortality, cardiovascular disease, number of lung cancer, types of cancer, arthritis, physical function, cognitive function, you name it, and we probably associate it. So about 16, 17 odd sort of metrics of aging or outcomes of aging or diseases or conditions of aging. And what we generally see and what we have here are the scaled associations. So essentially, they have been scaled within sort of one particular type of condition. What we generally see is that sort of the generation 1 clocks underperform as compared to sort of the second generation clocks. So one key takeaway I want you to have from today is that typically speaking, generation 2 clocks, and there's a reason why these generation 2 clocks were built, because they do capture aging in a better fashion as compared to generation 1 clocks, generally speaking, as you can see from this figure. But then the question is that which amongst the generation 2 clocks is the best? And I'm here like literally pitting one clock against the other. And the answer, in my opinion, is I think every clock is great, but at something specific. And so the reason why I believe this is that if you look at these different clocks, again, this data is in the same figure that I was showing previously just now, transposed, and we're looking at only 3 clocks. What we are really seeing is that certain clocks are better at predicting certain types of aging. So, for example, DNA GrimAge seems to be predicting cardiovascular and pulmonary aging much better as compared to sort of some of the other clocks. Or Dunedin pace seems to be predicting a lot of the outcomes of musculoskeletal and metabolic aging much better than some of the other clocks. Or BC PhenoAge seems to be predicting neurological aging relatively better, at least in this cohort study. And that goes back to the question that what are these clocks really trying to capture? All of these clocks are essentially trying to capture aging, whether they are trying to capture chronological age, time to death, pace of aging, whatever sort of metric that you're trying to predict, they all try to capture aging. But aging inherently is multidimensional. we don't all age in the same way. We don't all get the same diseases of aging at the same time. Some people will get cardiovascular diseases, others will get neurodegenerative diseases, some people will have more physical frailty as opposed to cognitive frailty. And that kind of hints at the fact that that generally aging is multidimensional. And what happens is that because aging is multidimensional, what the clocks end up capturing certain dimensions of aging or certain systems better than others. So take, for example, Grimage, which is a very powerful clock, but inherently it has a built-in smoking predictor, as well as was trained in the Framingham Heart Study, which makes it very obvious that it is for these reasons that it captures cardiovascular and pulmonary aging much better than some of the other clocks that we have out there. And this kind of bias kind of exists in every clock that is sort of being built at this point. And that is primarily because of what is going into these training models. And so one of the thought processes is that instead of having kind of clocks that are kind of giving a unidimensional score, why not just build clocks that are actually predicting that specific type of aging, whether that's brain aging, heart aging, musculoskeletal aging, immune aging, so on and so forth. And that kind of leads us to the idea of building sort of biological system-specific clocks, system-specific clocks that are hopefully more meaningful and giving you more insight into exactly how aging is happening within a system, and potentially even better for clinical trials, because very often a drug might not be targeting whole body aging. It might be targeting specifically cardiovascular aging or metabolic aging. And so what really needs to be done is then probably measure those particular types of aging, and you might see changes in those particular epigenetic scores as opposed to kind of the whole body aging score. And I'll talk a little more about these clocks in some of my subsequent sections. But I kind of want to just wrap up this particular section of my talk about sort of just the brief history of clocks, that we have known that there's been associations between DNA methylation and aging for a long time. And clock scores in general, at least generation 1 and generation 2, seem to be highly correlated. But when you actually look at the age deviations of these clock scores, these are the things that are really meaningful and distinct. And typically speaking, generation 2 clocks seem to outperform generation 1 clocks, and I say 2 plus as in 2 and Gen X included. And these generation 2 clocks seem to be capturing different dimensions of aging. And maybe what we need to do is kind of build clocks that capture these different dimensions more explicitly. And with that, I'd like to head into my next section, which is, what is the ideal tissue type that can be used to classify or predict age? And the answer to that, in my opinion, is blood. And I'll tell you why in a second. So again, going back to the history of clocks, there have been a bunch of clocks that have been built over the years, over the last 10, 12 years. And well, it's important to note how these clocks were built. So generation 1 and generation 2 clocks were typically trained in blood. So Panem, PhenoAge, Grimage, all of these were trained in blood, the exception being Horvath, which was trained in multiple tissues. But even though these were trained in blood, they were capturing an aging signal that was across the whole body. So if you look at sort of correlations or association between chronological age and sort of these DNA methylation age for a bunch of different clocks across a bunch of different tissues, you still see that they generally seem to be capturing an aging signal fairly well, and they are correlated between chronological age and sort of the predicted DNA methylation age. Of course, Horvath, which was the multi-tissue Horvath clock, which was built in using multiple tissues, seems to have the highest correlation in this scenario. So they are capturing sort of aging in different organs very well, but you still kind of need that particular tissue to do that. And then, of course, that some Gen X clocks capture organ-specific aging. So more recently, there have been clocks that use data from specific tissue data or tissue DNA methylation to capture organ-specific aging. Two examples here are PC brain age and DNAM cortical, which essentially use posthumous brain tissue methylation to calculate brain aging scores. And well, it has been shown that these particular PC clocks, PC brain age and DNAM cortical, seem to have a much stronger correlation with a bunch of Alzheimer's related outcomes or measures as compared to sort of some of the generation 1 and generation 2 clocks. Of course, the fact is that they are using posthumous brain data, which might be a little more difficult to access in most scenarios. So then the question is, which tissue is best to capture aging? Should we use sort of these organ level clocks, or should we use more of these whole blood clocks? And that goes back to the question, what type of aging are you really trying to understand? Are you trying to understand organ-specific aging? Then probably use that organ tissue and clock specific to that organ tissue. Is it general aging? Then use any organ. But since most clocks have been trained in blood, you're better off using blood as that particular in this particular scenario. But there are issues with these approaches. Well, to start with, that these organ or tissue specific clocks that essentially sort of require you to do a biopsy is often very challenging. You can't sort of do a biopsy of tissues in most scenarios. Like if you're trying to measure brain aging, you can't really take out a piece of your brain and do DNA methylation on it. That's really challenging. And potentially not really helpful. Of course, they capture organ aging very well, and of course, it makes sense that since they're capturing organ aging really well, they do not really capture whole body aging as appropriately. With sort of the blood clocks for general aging, the generation 1, 2, and 3 clocks or X clocks, they do not require specific tissue biopsy. They can be used from just simply blood, but they don't seem to capture organ aging as well, because of course they're trying to measure whole body aging. And as I showed previously, there is some amount of bias that comes into sort of capturing whole body aging as well. So what's the solution? And the solution there is that if we're able to develop a blood-based clock that can capture both organ-level aging and general aging, then we might solve all of these 3 problems. We do not require biopsies. We're able to capture organ aging from blood, which is an easy-to-access tissue, and hopefully kind of capturing different organ ages, we can create a more unbiased clock. And so the question is, how do we really do this? And this is what my research has been on for the last, I think, 2-3 years. We take whole blood DNA methylation and we use that to predict changes in biological system-specific biomarkers. So essentially, we group biomarkers into sort of mapping to certain biological systems. We do a lot of machine learning here, and I'm not going to go into the details of that because we're limited on time. But essentially, what we're doing is we're trying to track the changes in these particular organ specific biomarkers. And using those sort of proxies of DNA methylation proxies of these organ specific biomarkers, we sort of build system specific epigenetic scores, such as for brain, blood, cardiac, hormone, inflammation, immune, sort of 11 different scores, and eventually combine that into one unified score called systems age, which seems to be really good at predicting a wide variety of aging outcomes in a more unbiased fashion, I would say. Again, because of lack of time, I'm probably not going to talk a lot about systems age, the whole body clock, but focus more on sort of the novelty here, which is kind of these organ aging clocks, which I believe is kind of, in some ways, the real meat or the real value that this research has provided. So what we saw is that these system scores that we have built are very specific. And what I'm showing here is essentially I've performed associations of each of these scores with, again, a wide variety of aging conditions, and then scaled them essentially from the lowest score to the highest score having the darkest color, at the lowest score having like the lightest color and the smallest in text size. And again, this is in Women's Health Initiative, about 5K plus individuals. And what we see here is, for example, for cognitive function, the score which is obviously most associated is brain. For physical function, it's musculoskeletal. For total comorbidities, it seems to be inflammation that seems to be sort of most associated with this particular aging metric. For lung cancer, of course, it's lung. Leukemia, it's blood. For coronary heart disease, it's heart. For diabetes, it's musculoskeletal, but metabolic and inflammation come in close second. For thyroid disease, it's hormone. For arthritis, it's musculoskeletal. And we actually show this for 16 to 17 different conditions, that these system scores are very highly specific. And again, just to remind you all of this is being captured from a single whole blood DNA methylation blood draw. So all of these scores are calculated from just one single blood draw, and that's they're really specific. And when we actually compare these to sort of some of the presently available clocks, such as GrimAge, PC-PhenoAge, and Dunedin Pace, our system scores, or the most relevant or best system score, seems to be most strongly associated as compared to sort of the other clocks that are out there. And again, just kind of going back to the point that maybe capturing a organ-specific aging both provides more insightful sort of data, but also is better at predicting organ-related damage and organ-related disease outcomes. Again, to reiterate, all of this has been calculated from whole blood DNA methylation, which is why I believe that sort of blood is this one tissue that can be really used to capture aging across the body, as well as in specific organs. And the obvious reason for this is that it is one of the only organ systems in our body that kind of interacts with almost every other organ system. And of course, the ease of access of drawing blood samples makes it an even more promising candidate for sort of the purpose of aging research and gerontological care. And with that, I'd like to move into my final section, which is looking into the clock crystal ball, the future of sort of research and clinical applications for epigenetic aging clocks. And then sort of really well sort of puts this already, that what are the things that we require out of an aging biomarker? And I just kind of want to reiterate this. We want it to be responsive, that we have put in an aging intervention and we see a change in that biomarker. We want it to be predictive, where some level of change in that aging biomarker leads to an improved health outcome. And then eventually we like it to be surrogate, the holy grail, where an aging intervention leads to a change in the biomarker, leads to an improved health outcome. And I'd like to sort of go through each of these sequentially and just say that when it comes to predictive or prognostic biomarkers, I believe that aging biomarkers are kind of prognostic. I don't think we can call them prognostic flat out right now. And the reason for that is that in a lot of retrospective studies and cohort studies, aging biomarkers have been shown to be associated with future disease events. This is again data where we're seeing GrimAge and Dunedin Pace, and as I showed you all throughout my presentation, we have these clocks are associated with a bunch of disease outcomes, both future disease outcomes, present disease outcomes, functional disease outcomes, so on and so forth. But the challenge is that these are all epidemiological and population level cohort studies. We have little to no individual level studies that can really define healthy versus unhealthy ranges and quantify risk more like well-known clinical biomarkers. So take, for example, HbA1c, which has been cleared, we know exactly at this particular point is when we categorize as a person as diabetic, pre-diabetic, or not diabetic. Rather, HbA1c is used to define status of diabetes in itself. So aging biomarkers are nowhere close to sort of this prognostic capability at this point. But we can be, hopefully in the future, as we sort of improve on these biomarkers and perform more studies to show their prognostic behavior. when it comes to responsiveness, I believe that we are just scratching the surface here. And the reason for that is that there are a lot of clinical trials that are out there with a bunch of different interventions, lifestyle-based, pharmacological. Dan actually talked about one of them, the calorie trial, plasmapheresis, supplement-based. And the results for a majority of these trials, as you can see here, are not reported yet. So we do not know yet that which particular, whether these clocks are being responsive or not. What we do know is that for the limited results that in which we which we have gotten the results, or the results that we have gotten till now, there's no clear answer in the sense that, are these clocks really changing? Well, they seem to be changing in some scenarios, but I don't think 3 data points is enough to give a very valid answer on saying that, yes, I think we have responsiveness from these biomarkers. But we are seeing that there is responsiveness in some or the other scenario, at least in one clock or the other. So there's a lot more that needs to be done to show the responsiveness of these particular biomarkers. And then, of course, the surrogate biomarker is kind of our holy grail within the aging biomarker field. But to get to this point, we first need to show responsiveness and predictability, and only then aim for surrogate biomarker status. And I don't think we're anywhere close to it yet. It's going to take us some time, but hopefully we'll get there. And then there, of course, as Dan mentioned, that there are other issues, that we don't really understand what these clocks are truly capturing, and how does that relate back to the biology of aging? Is it upstream or downstream of the biology of aging? And again, all of this will be figured out as we understand more about these clocks and the underpinning of what's really being captured by these clocks. But I don't think all is gloom and doom. I just kind of want to leave you guys with a hopeful tale that these clocks have a lot of promise of personalizing aging research and gerontological care. And so I'm going to take a page out of my own research book and show you guys that how these aging clocks are really promising. So I kind of want to start with this, A Tale of Three Individuals, which is when we were analyzing our data in systems age, when we were building sort of these organ-specific clocks, we saw these 3 individuals that had the same chronological age and very similar overall biological age in a bunch of different clocks. But what was really interesting is that even though their chronological age and biological age seem to be very similar, their organ-specific age-accelerated or age-deviated scores were very different. They were aging in very different dimensions, as you can see right here. And that kind of gave us the hypothesis that maybe if they're aging in very different dimensions, they could also be getting very different diseases. And so what we did was, in one of our cohort studies, we clustered individuals based on their aging scores. And we saw that there were sort of 9 different clusters that showed up, which were sort of, again, this is age accelerated score. So there is these can be from any age range, from the youngest to the oldest in this population. And these 9 different aging clusters actually led to an increased or decreased occurrence of conditions or aging-related outcomes or aging-related potential reasons very differently. So each of these clusters here had an increased or decreased occurrence of disease or outcome or particular cause of aging. And what you're seeing here are the p-values, the Bonferroni corrected p-values. And so what we're really seeing here is that these clusters seem to be having very different aging patterns based on sort of the aging disease, you have. And just, again, there's so much you can sort of delve into this particular part of my research, but I kind of just want to highlight one particular point here, is that these 2 clusters, cluster 3 and 9, both had an increased number of cardiovascular diseases such as CHD, myocardial infarction, stroke, and And what's really interesting here is that even though both of them have similar outcome diseases like cardiovascular disease, they seem to have very different aging patterns altogether. Their organ ages are completely different, which potentially hints at the point that maybe these individuals should be treated very differently. So you imagine two individuals coming from each of these aging groups, they should potentially be treated in very different manner with very different pharmacological and lifestyle interventions. And hopefully these aging biomarkers that we are building will enable us to do that, will enable us to give more insights to our patients so that we can treat them and heal them better. And with that, I'd like to wrap up this section saying that in an ideal future, DNA-M aging biomarkers are used as surrogates in clinical trials. But to do that, we need to show their responsiveness and their predictability, both of which scientists are slowly making progress on. But that being said, we are already seeing potential value in having DNA methylation biomarkers in their ability to personalize gerontological care. And wrapping up, I just want to say that no clock is explicitly better except for when comparing generation 2 plus clocks versus generation 1 clocks. In blood is kind of the one tissue that holds, can be used to capture aging across both the whole body as well as in specific organs. And last of all, DNA-M biomarkers are not ready to be surrogate biomarkers yet, but we are seeing promising results, and hopefully in the near future, we will get there as well. And with that, I'd like to thank everyone in my lab who has helped with my research, my collaborators, all my funding agencies, and thank you.
All right, thank you very much. I think both of speakers gave us a really, really very, very good presentation. Yes, so there are several questions on the chat and Q&A. We have time for entertaining some of these. Why don't we start with the Raghav, since you just finished. There's a question about Nathan Price. He says, hi, responsive. Are these blood clocks to change in the relative levels of different cell types in the blood?
Yeah, so these clocks do change with cell types in the blood, and rather in at least some of our own research, I even try to model that into the data because cell type changes are actually meaningful. Think about immune cell types, right? In our immune clocks, we actually specifically model immune cell type changes and try to predict those using DNA methylation. The reason being that immune cell types, for example, changes are associated with a bunch of diseases and most importantly, cancer. So actually, I don't show this data, but the clock which is most strongly associated with or the organ clock which is most strongly associated with cancer is kind of the immune clock, which makes sense because if your immune cells are depleted or certain types of T cells are depleted, you will lead to having more risk of developing cancer because your body is not able to clear out cancer cells. So yes, these clocks are sort of responsive to changes in sort of cell types, and I think that is truly valuable as an option to have. Rather, some people suggest that we should regress out these effects. I believe that we should not, because we are capturing something really important about aging, even with these cell type changes.
Thanks. This question was answered already, but I think it will be interesting for the community. So this is about marketing. Yeah, so, and Dan, you want to comment on this?
Sure. I think, so this is from one of the panelists. I replied extensively in the chat, but let me, basically, there are a bunch of direct-to-consumer companies selling these clocks, and then there are companies that are selling clocks to healthcare providers, concierge medicine clinics, that kind of thing. This is this is a little Wild West, right? This is like selling nutritional supplements. The concern I have is related to the one that Raghav articulated at the end of his talk, which is that we don't, in the best case scenario for clocks like GRIM and PACE and maybe soon systems age, we have abundant validation data in population cohorts and a limited number of clinical studies that these things do what they're sort of supposed to do. But the extent to which they're appropriate for the kind of N of 1 clinical trials that the consumers of these direct-to-consumer or concierge medicine practices are running on themselves is an entirely different question. And I think that, provided somebody goes into it the same way people go into, getting their 23andMe profile, is kind of like, this is maybe fun, could be interesting, but I'm not going to take it too seriously. That's great. But when I talk to media about this, certainly the message I try to offer is that people shouldn't be changing their behavior in response to these test results absent consultation with a healthcare provider. And that a healthy dose of skepticism or circumspection is necessary in approaching any test results you get back. And I guess the other thing I'd offer is that these clocks, or the ones that predict health span and lifespan, were developed from whole blood. And some of the tests that are being marketed are not being marketed for whole blood samples. And the people selling the tests are not especially concerned, I think, with the ultimate validity of the information they're providing. They're concerned with the face validity, right? Does it look reasonable to the people who are getting it? And so, we all know that tissue is a really big deal, but the people who are buying these clocks may not. And so the fact that, this clock was developed and validated in blood, and now, somebody's going to compute it from a buccal swab, like, like how?
Yeah, sure.
And so, I'd love to see the receipts on some of this, but
Sure.
it seems like it's not doing a lot of harm at the moment.
Sure. Yeah. So what about you, Raghav? What's your opinion on this current? Yeah.
No, I kind of share the same concern as Dan here. So I've recently, I never used to use Twitter, now X, but more recently, over the last year, I've started using X, and I'm concerned. I see people, and I'm not going to name names here, but I see social influencers claiming about their youthfulness by measuring sort of some of these clock scores, and I'm flabbergasted by the fact that not only are they promoting their routines or regimens, but just based on some of these, and I think it's concerning because as good as these clocks are, and I being a researcher in this field, I'd like to promote them as much as I can, but at the same time, we're not there yet. We're not there where a single N equal to 1 is enough to sort of build interventions.
Yeah. I know Ron has a hand up. Just hold on. So Raghav, while you're on the mic, so what do you think about the application of AI in this? Obviously, this got to be, all the precision medicine and all of those you're putting. What is your thought on those?
Yeah, I think so. I've been doing sort of some form of AI for the last 10 years now, and what I believe is that it's already embedded. ElasticNet is a form of artificial intelligence. It's the most trivial form, but it's still artificial intelligence. And I think we're getting better and better at including sort of more of these AI routines into our into both biomarkers, but also sort of using that personalized care. Like, for example, the last section that I showed you about my research, it's in 2,000 individuals, and I think it's a very small subset to actually drive a bunch of insights. I don't think there are 9 aging clusters or 9 aging subtypes. I think there are potentially way more than that. But I do think that if we have the right amount of data, right, and the right machine learning models, we can actually get there. We can use these AI tools to create sort of these more personalized interventions, as well as more personalized understanding of aging. Again, as I say this with a certain amount of like grace that needs a lot more work, a lot more data to get to that point. But I think we're getting there.
Sounds good. Ron, go ahead. Unmute yourself. Ron, you seems like you're frozen. Okay, while he's getting back, let me read the question from Josh. There's a question about for the bespoke aging clocks that predict skin cancer and cancer health. Are they being used to predict trend on non-pathological phenotypes like wrinkles or skin elasticity? Do you know?
I mean, can I just insert something in the question here?
Yeah, go ahead.
I feel like somehow, and Raghav, maybe you have a different take on this, but like somehow the word clock became synonymous with the word algorithm. And like, so I don't know in the case of these particular clocks, whether they are designed to predict age-dependent risk for skin cancer, or they're just a skin cancer risk biomarker that we're calling a clock because it weights a series of inputs into a prediction of some outcome. But I think that it would be helpful if those of us who are trying to measure aging played some defense, or maybe we just need to give up and change the word that we use. But like a clock and an algorithm ought to be different things.
Okay. Ron, you're back. Go ahead.
Yeah, thanks. So for Raghav, excellent talks, both guys, and not surprised by that at all. What about directionality or bidirectionality when you're looking at sampling from the blood and looking for speedometers in different tissues? So is it aging in the blood that's driving aging in the tissues, vice versa, or a combination of the two?
Honestly, Ron, I don't have an answer to that. I wish I did, because I think that's kind of the challenge with most of these epigenetic aging clocks, that we do not understand the directionality. There are two hypotheses here. One hypothesis could be that this epigenetic aging program that's across all our systems, and it's even programming for each of our organs aging in different rates, and that's why we are able to capture sort of these changes in blood, because it's across the whole body, and it's valid, and this particular epigenetic aging program works across all tissues. And so potentially, even if you were to take out this DNA methylation from a liver, you might still be able to capture brain aging. That's one hypothesis. The other hypothesis could be that there is actually some flow of information, right? That DNA, these DNA methylation is being modified by potentially other molecules that are coming through, say, the liver or the brain or the heart, and that's essentially what is modifying these DNA methylation markers in the blood, which to me seems like the more obvious hypothesis. I'll be honest, that is what is essentially happening and why we are able to capture these organ-specific aging in the blood. But I think it needs to be tested. I don't have an explicit answer on which of these might be the scenarios.
I think you laid out what the options are better than I could, for sure. Let me just ask a quick question of Dan. When you were looking at weathering, which I think is one of the things that comes out of study of poverty, right? I don't remember who coined it, but what about the outliers? I'm pretty sure you've looked at the outliers, those who have whatever the level of poverty is, but they seem to tolerate it better or worse.
Yeah. So Arlene Geronimus is the sort of mother of the weathering hypothesis. And I think we don't have necessarily, we don't have answers about what makes some people resistant to the depredations of adverse environmental conditions. I think, there's a really creative strategy here that Morgan actually introduced in a paper she published in the Journals of Gerontology some years ago, in which she conducted a GWAS of longevity she conducted a GWAS of long lives in smokers. And it was a tiny sample, and it was sort of an exploratory analysis, but it gives us a suggestion of a way that we might use some of the data that already exists to try and identify molecular features that signal resilience to these really damaging environmental exposures. And to date, we've not seen anybody essentially build clocks in the opposite direction. So we're kind of focusing on aging fast versus slow, but who's surviving when they shouldn't be is a really good question if what we're trying to do is build geroprotective therapies. And so, that's definitely something that we should probably all be doing more of. What I will say in our own work is that certainly for kids who are born into adverse environmental conditions, it's not destiny necessarily. For people who across their lives rise out of poverty, we see outcomes in aging metrics, epigenetic clocks, blood chemistry clocks, other kinds of metrics that are quite similar to people who had very different early lives. And that gives us some hope that interventions to change people's environmental conditions can actually change their pace of aging. And in fact, I was looking for a way to sneak this into the conversation. Andrea Baccarelli and I have a new review paper out in Nature Aging last week, in which we make the case that we should really be thinking about the environment as an opportunity for population scale geroprotection, that by changing the places that people live and the ways they live in those places, we can slow their pace of aging and build healthy lifespan. But that's me pivoting off of your question. I think what you're really highlighting for us is that we need to spend more time digging into how some people manage to survive environments that kill most of their peers.
Thank you.
Thanks. Certainly, resilience is an important issue. So, let me give it one last question, and we'll end. So, there's a question about the variability in individuals. So, again, it's certainly, we are affected by environment and also exercise and all. Related question is, can it reversed? What do you think? Quick answer, please. Have you tried it? How much variability in individual, Raghav, in your experiment?
No, so I've not really seen if there reversibility per se. That's again something that we need to look into. But I think
what about the variable? What about the variability in single individual for a short period of time? Have you done any?
So there are a couple of things. I think there's one part about reliability, which I think Dan mentioned about, that if you do like replicate studies of the same individual at the same time, there can be a lot of variation because of like the methylation sites you choose. But even within an individual, if what we have seen is that, for example, if you were to do a buccal swab and then brush your teeth and then do a buccal swab again, you'll get completely different epigenetic ages.
Right.
And so there is a lot of variability, and of course, for something like even like the circadian cycle seems to affect some of these epigenetic clocks. So if you were to capture DNA methylation blood from morning as opposed to evening, you'll get very different results. Something else that seems to affect is your glucose fasting status. So at what point? So that again, there is individual variability as well, and which is why kind of we go back to the point that the n equal to 1 is we're not there yet with the n equal to 1. You might, if anything, what you need to do is probably do 3 tests in one day and then take the average of that, and probably that's going to be a better measure than just saying one particular blood draw, one particular glucose.
Thank you.
Yeah,
go ahead.
I think it's a really important question, and one that NIH is in a position to maybe do something about. But we actually don't know much about day-to-day variation, week-to-week variation in these metrics of biological aging. And it is a question that we ought to know the answer to the extent that we are deploying them as endpoints in clinical trials, or considering making some ruling on whether we think it's a good idea or a bad idea for people to test themselves. And the barrier to knowing the answer to that question is essentially that it's still pretty expensive to measure these clocks. And so to conduct a weekly study of several dozen or 100 people is quite costly, and is the kind of thing that might require some funder to step forward and say, we think we should know the answer to this question, so somebody should do it for us.
Well, thank you very much. Sounds like this is all important issues that we need to overcome in the later years. So thank you all for the attending the webinar, and I think this was a very successful webinar. Siobhan, you want to say for the next one, or?
Sure. And thank you both for outstanding talks. You really covered these complex issues in such a nice way. We will meet again on December 14th for our next webinar, same time, and we'll look forward to sharing information with all of you. And again, thanks to everybody who joined and to our excellent speakers.
Thank you very much. Thank you. Great job, Dan and Raghav. It was great.
Thank you.
Bye-bye.
Bye.